All right, so this is Unit 6, and this will be the last content that is on the AP exam for this year. I'll do slideshows for 7 and 8, but this will be the last stuff that will actually be tested on. So the first stuff we're going to talk about is DNA structure and function. We talked about this quite a bit, so hopefully this will be easy, so I'm going to go pretty quick. In our bodies, we have a cell and has a whole bunch of squiggly chromatin in it, and then whenever we organize that, condense it, we call it a chromosome. So it's just vocab, and we've talked about that before. Each chromosome has 2 sister chromatids. They are attached together at the centromere. Remember the weird thing that I was like, mere, and then they connect together. Kind of a weird thing to remember that. And then this just shows you an image of that. So whenever chromosomes organize themselves, they condense and wrap around little protein-like structures or protein structures. And one of the things that they wrap around is called a histone. I think of a histone like the spool, and the DNA is like the thread, if you've ever seen like a thread of a spool of thread. And it just helps it wrap onto something and then be able to coil up and then condense into that shape. Just a picture, pretty cool. And then this just shows you sort of from what we think of as a double helix up into the structure of a X-shaped chromosome, how they wrap and coil and condense. The little purple spheres like next to the little number 5, those are histones. Then for humans, we have 46 chromosomes. It's called a diploid number. It's called 2N. When we take a picture and organize them according to shape, length, banding, location of the centromere, it's called a karyotype. And this is just a picture of one. So this would be a picture of a female. And then if we go and we scan through a little bit, you can see that as far as numbers, her chromosomes look like she has the normal number of chromosomes. And then we have 22 sets of what are called autosomes and 1 set of what are sex chromosomes, so the X and the Y. And then this is just showing you how the different things might come together. And then we might get a daughter or a son. And so it's not great because when it converted over, it didn't do it right, but I think you can figure that out. Watson and Crick, gross, get credited for figuring out the structure of DNA. Because they put all the pieces together and they published the paper the fastest. But as you know, as I've talked in class, they are not my favorites, especially James Watson. And then the pieces and parts that make up DNA and RNA, the little bits, they're called nucleotides. There's 3 parts to a nucleotide, a 5-carbon sugar, a phosphate group, and then 1 of 4 nitrogen bases. And then they're called pyrimidines and purines. That is a chemistry structural kind of thing that you don't need to really remember, but that's 2 of them. Thymine and cytosine are pyrimidines, and adenine and guanine are purines. And then you have a pyrimidine and a purine pair up, and then the other pair is the same type of combo. And then this just shows you what they look like. They have they kind of look similar to each other, like adenine and guanine kind of look similar, and thymine and cytosine kind of look similar to each other. But the reason that they are shaped specifically and then they partner is because of the bonding availability sites on the structure, and then they match with the bonding availability sites on the corresponding or complementary nucleotide, and then they can bond up. The fact that there's 4 and they bond up not exactly perfect is why there's a kink in the double helix. It's not like a spring, like exactly perfectly like a spiral. It's like a tilted spiral, and that's because of the structure of the 4 nucleotides. And then this is talking about Chargaff. He figured out that there was the same amount of adenine as there was thymine, and then there was the same amount of cytosine as there was guanine, and then he presented the fact that they must partner up, and he was right. So that was a big hint as to what paired together, and then a lot of sort of other discoveries came from that. Rosalind Franklin is my favorite. She took a picture of the DNA molecule, most specifically down through the tube, down through the spiral, and it gave a certain image. And her technology was called X-ray crystallography. She shared that, or her finding was shared with Watson and Crick, and they thought it was a triple helix. They couldn't quite figure out how all these things equaled each other, and then it would how it would be structured, and hers was sort of that last little piece of information that they used in order to get that. They got credit because she died, but we've talked about that extensively in class. And then this just talks a little bit about how they're bonded together. They're the A's and the T's and the G's and the C's are held together in the middle by hydrogen bonds. We've talked about hydrogen bonds are not really a bond like a covalent or an ionic bond. They're more of an attraction, and that our body uses hydrogen bonds a lot because they are easy to break and easy to form. So it doesn't take a lot of energy. Every time we want to make a new cell, if it was like an ionic bond, that would take too much energy to break those things apart. So our body has evolved into using hydrogen bonds a lot because of, oftentimes, because of the energy necessary to bond those or break those bonds. And a picture of the people that I don't like. Gross. And then this talks a little bit about Rosalind Franklin. She was not given a piece of the Nobel Prize because she died when she was 37, because when she was doing her research, it used x-rays, and back then they didn't know about the risks. So she took no protections. She ended up getting cancer and then dying. So, and you can't get a Nobel Prize when you're dead. It's called posthumously. Fun fact. And then that's a picture of her. We love her. And that's a picture of her down the center of the tube of DNA picture, her X-ray crystallography picture. And then this talks about that the A's go with T's and G's pair with C's. And then RNA is a little bit different in five big ways. One, it's usually single-stranded. There's no thymine, there's uracil. It can leave the nucleus when DNA cannot. The sugar is ribose sugar. It's why it's called RNA, and instead of deoxyribose sugar, the difference between those is deoxyribose sugar contains one less oxygen than ribose sugar. It's deoxide is why it's called that. And then we can make little chunks like messenger RNA, and then other kinds that we'll talk about in a bit that we use in transcription. Replication is making a copy. And so we have to replicate DNA because it can't leave the nucleus. And so we have to make a copy that then we can kind of destroy and use for transcription so that we can get it out like to the ribosomes or out into the cytoplasm, and so that it can do the job that it needs to do. And it's also when we make a copy to divide up, and it's the same process that we divide up to make like a second cell for mitosis and stuff like that. There are a lot of enzymes, but these are the, there's three that I really want you to remember, and then that fourth one, just sort of an extra one that we don't talk about very much. But DNA helicase is a big one. It breaks the hydrogen bonds between the strands. The joke is it unzips the genes. And then the next one, topoisomerase, is we break the bond, but then we have to unwind the helix. And so that unwinds it. So it's sort of a structural break and then a unwinding. And two different enzymes do two different jobs. Polymerase is one that ACE is a enzyme. Polymer is a structure. DNA polymerase is the enzyme used to make the DNA into a polymer. And so if you're going to make something from a single thing to a many thing, you have to add parts on. And so DNA polymerase is the one that takes the nucleotides and then puts them onto the end of the strand and then adds it all together. And then ligase is the one that that like a ligament connects. It's the one that connects the stuff. And then there's DNA polymerase 1, there's DNA polymerase 2. They add things on the end. They proofread to make sure that the job got done correctly. So there's different ways, but they all have to have a little signal. And I showed you a couple videos in class, so if you wanted to watch them again. It's a little like, hey, this is where we're going to start right here. A chemical signal that is put there and that our body chemically recognizes as where it's going to initiate, and so it's called a primer. And then this talks about when we watched the video, it was called the leading strand and the lagging strand, or the crash course guy is kind of funny and he calls the lagging strand the scumbag strand. So we can only add, and if we have our 5' to 3' ends running anti-parallel to each other, excuse me, we can only add on to the 3' end. So the 3' end, they just pop on and everything's happy and everything's great. But on the other one, it's sort of like, it's like a back stitch on a sewing machine, or you go up a little and then you have to go back and do another little section, and then you go back a little further and then do another little section, and then the polymerase and ligase zing through and connect everything together, and then everybody's happy. And then we do, it's called semi-conservative DNA replication. We replicate semi-conservatively. So if we have our original strand of DNA and then we split it apart, we join nucleotides here, we join nucleotides here. So these two are half. Semi means half, and these are saved. And so they're used as a template. And what that does is it gives us a higher chance of accuracy because we use an original template to make the copy. And then this is just a picture that talks a little bit about the different enzymes and where they work. This is just an overview. The DNA goes through transcription to make messenger RNA, and then messenger RNA goes through translation to make a protein, but we'll talk about both of those. Transcription is when we make a copy of the DNA that then can go out and into the cytoplasm to do a job. And so we have ATs, Gs, and Cs on DNA, and then we have a little piece of the DNA, the gene of the DNA. So say we ate some, it's the example I always use in class, say we ate some cheese. And so we have lactose in our body now. We're not going to constantly make lactase if we're not eating or drinking like stuff with milk or dairy products in it. So when it's there, we make it. And so our body has to say, hey, this is here. And so that comes through cellular communication and going down through the cascade, and then we have a product at the end of that. That product, when we talked about that in the previous unit, is called a transcription factor. That transcription factor goes onto the DNA and says, this chunk is what we want. This chunk contains the coding for the enzyme lactase. And so it's like, okay, time to start. And so then we transcribe that little part, and then it can go out into the cytoplasm, and then it can make the enzymes, and then we can digest our milkshake, and then we are so happy and great. And so this just talks a little bit about that, about the enzymes, and that, and I talked a little bit about what makes DNA and RNA different just a bit ago. So there's three kinds, two that we really focus on, one that's kind of a structural thing more than a helps a lot in doing a job. So messenger RNA takes the message from the DNA to the ribosome. And then ribosomal RNA is it's a couple of subunits that make up structurally the ribosome, and that it helps to our body to go through the process of transcription and translation, but mostly it's structural. And then transfer RNA is a little sort of floaty around one that has 3 little letters and then a little amino acid on the other end, and then it sort of brings it in and pops it onto the little thing, and then pops it on, pops it on, pops it on. It transfers the amino acid to the ribosome, which then gets added on, and then we make a protein. So we'll talk about translation. So translation is taking that messenger RNA and translating it, turning it into a protein. And so this is, I talked about this a bit ago, but gene expression is when we take the gene, so we have the gene for lactase, and then we express that gene and make the lactase. And it has three, so we'll talk about, but that'll be in a second. And then same thing, RNA polymerase makes RNA into a polymer. So it's just the same process, but with RNA. And then same thing, I talked about this earlier. And same-ish kind of thing as a primer, but we need a little section to start things. And so, and oftentimes it has a lot of different parts that have to come into contact with each other. So our DNA can bend, and it can touch in different places, and then it can make different things, which we'll talk about in a bit. But it is a place where has to be sort of turned on or turned off depending on what it's making in order to work. And then again, we talk about the 5 to 3 prime. It unwinds. We read it, we add the free nucleotides, and then we make the messenger RNA. And then this is how we regulate how things sort of get made. We have, let me back up a teensy bit. Back when scientists did the Human Genome Project, they thought there were going to be so many genes because we make so many things. And what happened is they figured out that it's not really that much stuff in there. So when there's not very many kind of instructions, then our instructions have to be able to be used in different ways. And so introns and exons, and then how things fold and sort of touch each other, things getting turned on or getting things turned off, is all part of we have a really minimal kind of mass of stuff that we can do a whole bunch of stuff with. So it's a really cool evolutionarily how we didn't make a bunch of stuff, but we can do a lot of stuff with that that we do have. And so introns are the part that are in there that eventually get taken out. And then exons are the parts that get expressed, is how we watched a video and they had us, and I thought that was a great way to remember that. But the introns are parts that don't get coded for. They're filler. Sometimes if we have DNA, let me do it like this because then my hand will work, and it has to fold so that this part of it and this, wait, with my hand, so this part and this part have to touch, then this part is junk. And so it's just like, it's like available length to help us stretch around. And so so that these two parts can get attached to each other, this sort of has to be there or else it wouldn't work. So hopefully that kind of makes sense why introns are there, but we don't. But then some people say they're useless or they're called junk or different stuff like that. They're actually super useful in sort of the flexibility and the multi-uses of a strand. And so this just talks a little bit about alternative slicing, that we have a minimal amount of stuff, but we can make a bunch of stuff. And then when we take out the stuff that we're not going to translate and we keep all the stuff and we sort of squish it all together, then we put little chemical signals on each end. And so there's a cap on one end and another cap on the other end, and they just sort of signal the body to say, this is where we're starting, this is the end of it, and then we know, which is a cool thing that it just boggles my mind sometimes. And then this just shows sort of a visual of that thing. And then this is just talking about how we make the protein from the messenger RNA message. And so at the ribosome, the transfer RNAs along with the message of the messenger RNA all come together. And so on the messenger RNA, there is a start codon, and then after that, every set of 3 letters is called a triplet or a codon or triplet codon. Sometimes you'll see, and each triplet set of letters gives us a specific amino acid. And so this just talks about what there are. AUG or methionine is the start codon. It's the molecular message that says we're starting here. So like say we ate some cheese like before, and we have our lactose, and that lactose comes in and signals our cell, and then we go through the cascade, and then we have the transcription factor that pops out, and it goes to the DNA, and it says we're gonna make this little chunk into a messenger RNA. That little messenger RNA scooches out. Well, the beginning of that messenger RNA, the beginning of that message starts with an AUG start codon, always. And so that's how our body knows that this is where we're going to start. We're going to make the protein, the enzyme, lactase. And then there are 3 chemical signals that are called stop codons. So when our body sees that little combo doesn't code for any amino acid, it just is the chemical signal that our body says, okay, we're done, and it breaks off, and then it leaves. And so this is there's several ways that you might see the genetic code, but on this one, there's a left, the top, and then the right. So if I give you, say I give you CCG, and so if we look at that, we go to C on the left, C up at the top would come down to that box, and then G we would go over, and then it would be CCG, and then pro is proline, and so that would be the amino acid that little combo would code for. This has little stop signs for the stop codon, and then the AUG, if you can find that, it says MET right next to it, which is not an amino acid, it's just the chemical signal for it to start. And then this is just a list of our amino acids. And then this is just a picture of sort of that going through the process. And then, so if we have our 3 on our messenger RNA, 3, 3, 3, 3, 3, 3, 3, 3, 3, all those, every one of the 3s are called codons. The corresponding complementary one that's on the transfer RNA, they're called anticodons. And so they just structurally kind of look like a T. Sometimes people think that it looks like that, but they have like a weird little structure, but they have 3 little letters and then a amino acid on the other end. This is an example for tryptophan. And so this is just talking about how we take that message and we make it into something. There's 3 steps for translation, which they're pretty easy. It's almost exact. I think it's exactly what the words sound like. Initiation, elongation, termination. We're going to start it. We're going to make it longer. We're going to stop it. So, which is exactly what we do. We have a start codon. We add a bunch on as long as the messenger RNA gives us a message, and then we end it when we hit one of the 3 stop codons. So hopefully that makes sense. And then this just talks about those things, that it's initiated when it's told to start. It's told to start because it sees AUG or methionine or the start codon. And then elongation is it just keeps adding them on until it's supposed to. Some proteins are super duper duper long, some are super long, but they're all pretty long, but some are huge and some aren't. So it just depends. And then you hit that stop and then you're done. And it breaks stuff off, and then the protein goes out into the cytoplasm. Maybe it gets packaged up and it gets sent outside the cell. Maybe it gets used up in the cell, but whatever job it has, that's the job it gets to do. And then sometimes they can do the process at different spots along the strand. And so it makes it really, really efficient because it's like a whole bunch of factory workers kind of working on different parts of the car, and then all of a sudden the car is done, which is cool. And then this talks about how we control genes. How do we turn them on and turn them off? Sometimes we get a signal, and sometimes we use other things. And so one of the things that makes like animals and plants and higher level organisms different and fancier than lower levels is differentiation. And so we have we had 1 egg and 1 sperm, and then it came together and we have all the chromosomes that we need. And then we think about mitosis. And so we would have like a blob of the same little cell. Well, that's not what happens in our bodies because of differentiation. And so we have special controls right at the beginning of development that make what would seem like a little blob of nothing that should not really turn into anything fancy, turn into something fancy. And so we can turn things on and turn things off, and then we have special things. This is just sort of a generalized overview. And then this is what I was talking about earlier with we can turn things on and turn things off, or things have to sort of bend and meet, or we have processes that keep things turned off because we don't want them or because we don't need them, or on for the opposite. And then this talks about epigenetics super quickly, and we didn't really have a bunch of time to do this semester, but that's the way it goes. Methylation, so on this it says enzymes that acetylate histones encourage transcription. Adding a methyl group to a histone prevents transcription. And so if you add, because of environmental factors, because of behavioral things, different things that we're sort of still studying with epigenetics, but they have found that if a gene is passed from one generation to the other and there's an acetyl tag on it, that makes it so that you see it. Methyl shuts that process down. And so say there's addiction in your family and your mom might say, well, you know, Uncle such and such, he drinks a lot, and you might have that gene, better make good choices, then you can do that. And then maybe you'll get methyl groups, and then you wouldn't get the gene expressed, which is pretty cool. Okay, and then this is talking about how we know where to transcribe things or start or stop or all the signals. We talked about that. Another cool thing that our body does is RNA interference. That video link is we watched in class, and it talks about trying to make a really, really, really purple flower, and that it ended up making a white flower because of our body's sort of immune response to viruses. It sort of sees that as foreign, and then it's a cool thing that happens with sometimes is a protective thing, but it's called RNA interference. So if you forget what that is, maybe watch that video. And then after we translate it, sometimes things can get changed. And so a couple cool things that happen is it's sort of like, well, how do we know how to make all the stuff if we didn't have any stuff made? And where does this come from? And it's sort of the chicken or the egg kind of thing. And there's certain things that are called master genes. And they are the ones that sort of are like the masters, just like it sounds like, that sort of help the process get started. Pattern formation is, when we talked about, and we didn't talk about it a ton because it's in Unit 7, but it talks about sort of like we start with a blob of cells, and then it starts to develop into like tissue-y stuff, and then bones, and then like hair, and chickens and humans and turtles and everything. They go through that progression, that pattern of formation in the same pattern, which is a hint as to common ancestry, because we develop in the same way. But we develop away from just a blob of cells because of different genetic material that tells us how to get to be a human or to be a chicken or whatever. Pretty cool. And then this just is showing the same information just in a picture. And then we have, this is just a extra slide, which doesn't give us too much information. A cool thing that happens in females is we have two X's. And so a lot of times in mammals, we have what's called X chromosome inactivation. So we have one that sort of leads the way, and one that doesn't. Sometimes we get a weird thing that happens in cats, and we talked about different examples that we kind of get a weird patchy thing. Or one of the other examples that we learned about was like sweat glands, and that they're sort of patchy on some in certain things. So it's just cool. So that's a cool thing that everybody does. And then this just talks a little bit about the same stuff. There's a little video that if you want to watch that, it just talks about that with bar bodies and activation and mosaics and stuff. And then this talks about how when we inherit two X's or an X and a Y, when we develop into a female or a male. And so males on the Y chromosome have a specific master gene. It's called the SRY. I always joke and say, ha ha ha, sorry, you're a guy, because, you know, that's how I am. But when you lack that gene, you turn into a female. And so that's how we turn into what we turn into. And then prokaryotes are a little bit different. They're a lot simpler. That's why we use them to do like research and genetic modification and all that kind of stuff. And so they have parts. We watched some videos on these with the lac operon and the trip operon. There are parts that you can turn on or turn off in order to make what you need or not what you need. But it's the same sort of idea, just a little bit different when it comes to prokaryotes. And then this is just specific examples. And then more stuff about that. But this is a lac. And then a little bit about mutations. We've talked about this a couple of times, I think. But polymerase does two things. One, it adds spare nucleotides onto the end of the sequence. It also makes sure that we copied it correctly. So sometimes that doesn't work. So we get a mutation. And so this is just a picture of a little bit of weird breaks that shouldn't be there. And then it shows damage, and there's lots of different reasons for that. So we have different kinds of radiation. It's pretty easy to understand, but they cause damage to the DNA. I talked about in class that in America, we have a higher incidence of skin cancer on our left arms because that's the arm that's by the window. When you're driving the car, it's the opposite in countries that drive on the right side of the car. I also showed you pictures, I think, about like people, like if they sat next to a window for their entire career, like they worked at a little office and like they had like wrinkly skin on one side and okay skin on the other side. But you can Google all that. So just mutations, and we've talked about that, and you know what that is. Just a little change for whatever reason. And then this just talks about how they get made or not made with different products, amino acids, proteins, all that kind of stuff. And then this is specific examples of some mutations that we can have. So, and they sound pretty much exactly what they are. A base pair substitution. We took out the one that was supposed to be there and we put in one that's not supposed to be there. Or deletion is we took out a chunk, or insertion is we added an extra chunk. And then hemoglobin is a pretty important, carries oxygen in our body. And it is if we think about a primary, secondary, tertiary, quaternary, it is a quaternary because it is several different things stuck together. But it carries oxygen through our bodies. And when it isn't made correctly, because there's mutations genetically, then people have anemia. A specific example of anemia that affects the hemoglobin is sickle cell anemia. We talked a little bit about sickle cell, but it's when our cells, our red blood cells, sickle. Instead of being a nice like disc shape that flow and carry oxygen and everybody's happy, they sickle into like a grim reaper, like a sickle. And then those don't flow nicely and they get clogged up and it's painful and there's decreased oxygen carrying capacity. There is pain in the parts of the body where that gets sort of jammed up in the red blood cells, or I'm sorry, in the blood vessels. And then it can be really, really dangerous. And then this is just an example and a picture of those cells is This is T-Boz. She's in TLC, and she has sickle cell. A lot of people do. A lot of people that are of African descent, and some other, but a lot of it is African descent, and it has to do with places that have malarial risks. So they have a decreased oxygen carrying capacity. Their cells might sickle, but they can't get infected with malaria, which is really nice if you live where there's malaria, but it's not really great. And it's not too super useful if you live where there's no malaria. And so this is another one that if you take something out or you put something in, it's called what's called a frame shift. If I take a chunk out and this shifts down, that's just what it's called that. Or if I shove in some extra stuff, it shifts it down, and then the sequence of message, the codons are shifted, and then they're wrong. And then this is just talking about different things that cause that to happen. So it's just a little bit of vocab. And then we talked a little bit about biotech. So this was restriction enzymes are the ones that cut. And we did a big packet about this. And so we can cut, and they make like a little like shelf or like jaggedy part. And so the sticky ends is what we call them, have to be complementary in order to recombine with something else, maybe a bacteria and some strawberry DNA or stuff like that. And so whenever we combine two things that weren't originally together, it's called recombinant DNA. And then this just shows how that's done. And we did, like I said, we did the activity on this in class, so this should make sense. And then we are learning more and more and more where genes are located, so we can go in with certain restriction enzymes, cut the chunk that we want, make a bunch of copies with polymerase chain reaction, and then use it to do research or modification or crops and agriculture and stuff like that. So it's a lot of cool stuff happening. The genome is our genome. If you Google stuff, we know pretty much what the human genome is now. But now they talk about the proteome, which is the making proteins from that. That's sort of where we're having some trouble. It's not as easy as it may seem. I don't think it seems easy, but that's where we're sort of stuck. And then we have libraries that contain things that we figured out and other things that scientists can use. And then whenever we want to find something specific, we can tag it, and then it can be seen and we can find it, and then it can help us to do different technologies. Polymerase chain reaction is just, we talked about this a little bit, you heat it up, it separates it, the spare nucleotides come in, they cool it down, they attach together. You heat it up, they spread apart. It's the way that we made copies. It's like a lab version of replication. And then this just talks about how PCR gets done. If you Google it, it's pretty simple, not simple like scientifically, but conceptually to understand what's going on. And then this is how DNA sequencing is when, like the human genome, or when we sequence an organism. And so they put like tags, chemical tags on all the A's, chemical tags on all the T's, the G's, the C's, and then it sort of glows like green and blue. Like all these are green, those are A's. All these are blue, they're T's. And so they glow, and then they can say the sequence by the machine. Oftentimes determines the color or the sort of signal that the probe or the little tag is sending out. This electrophoresis, we also did another super simple thing about this, and then we did a graphing packet about electrophoresis. The heavier something is, the less it can travel because it's too heavy. And then the smaller something is, the easier it travels, so it goes down the gel further. We chop it up with restriction enzymes. We make a bunch of copies with polymerase chain reaction. We separate it into the desired chunks using electrophoresis. And then a cool thing when we do electrophoresis is you can make like a DNA fingerprint, or you can chop up different strands of DNA and then use it for like a crime lab. And there's really cool thing that is super unique to each person is called a STRs, or short tandem repeats, that are unique. It's like a DNA fingerprint for each individual person. Now, the only bad thing is if you're an identical twin, then you can murder somebody and your twin could, get proven guilty. But other than that, you're probably going to get caught. And then this just talks a little bit about that. And then genetic engineering is when we take a little bit of genetics from this and a little bit of genetics from this, and then we put them together, and it makes hopefully something better. There's controversy that goes with this because it's sort of tinkering with genetics, which can be scary. But a lot of it can be really, really helpful if we can make drought-tolerant crops, or if we can make crops that make their own pesticides versus having to spray the crops. If we can make crops that live longer because they are tougher against bugs, that makes more food that they can feed more people. I think that's a good thing. Some people disagree with me, and that's okay. And so this just talks about some cool things that we've been able to do. It talks about jellyfish and insulin, and there's so many examples, and I geek out about it a little bit, but pretty cool. And this just talks about different possibilities that we can do with genetic modification on crops. If you're into agriculture or like farming or stuff like that, you might know about this stuff. If not, you can learn about this stuff. It's super interesting. And then this just talks about the controversy. Whenever you fiddle with genetics or combine things that shouldn't naturally be combined, it can be scary. And sometimes it's dangerous. And so we have people that disagree with any of it, people that are all for it. Some people are like, well, I'm not really sure, but making insulin sounds good. So, you just got to figure out or be educated about it. And that's why I talked about this is with my students, is because this is new sort of technology and the ability and transgenic crops and all this kind of stuff. You're going to have to probably make some decisions, like when you vote, whether you agree with it or not. A few years ago, they wanted to get foods, I think it was if they were genetically modified, that they had to have a label on them stating that they were genetically modified. Well, realistically, most food at this point, especially if it contains corn, is genetically modified. It just is. And so what happened as a result of that, it got voted down. They weren't required to do that, but non-GMO food manufacturers, they started to put a little label. It's a little square. And so if you eat organic or non-GMOs, you probably know what I'm talking about. But if you're curious and you look at stuff like that, like go and look, and then it's there. It's cool. And then this is just talking about using different genetics in animals to make different products that we might be able to use. And so this is just some examples of like clotting things or different things in milk, and just so much exciting and cool stuff. And then just some examples. I thought those pigs with two colored noses were funny. And then a chicken with no feathers looks really ugly, but hey, when you're in a chicken processing plant, looks pretty good. And then this just talks about dangers and concerns and different stuff like that. And then just some things to think about. And then this is sort of what I was talking about with like voting and like genetically modified humans. Think about sports, think about intelligence, think about things like that. Like, it's really awesome if I can genetically modify and get rid of cystic fibrosis, but what if it turns into, let's genetically modify to make your child taller or smarter or whatever? It can get a little bit scary. And then cool things is gene therapy is the good part of genetic modification and genetic treatments. And so there have been a lot of big strides, especially with cystic fibrosis. which is really cool, that they're able to genetically change stuff and then help certain groups of people. And then eugenics is sort of the dangerous part of it. It's like making the perfect human. It was started in Cold Spring Harbor Laboratory, which is in New York, but it is where Hitler got his ideas for the perfect race. And then it turns into who decides what's beautiful or who decides what's good, and then it sort of gets scary. But when you try and make the perfect race, that is if you ever hear eugenics, it's not really a good thing. And James Watson likes it.